Good day. We're on the fourth page of your genetics notes, and I've just filled in the starting point for the Punnett squares. I went big T, and I put them here, and I put little t, and I put them there. And that's showing the meiosis. This is the segregation, the law of segregation, that these genes segregate out during meiosis, the formation of gametes. Here I went big T, and then I put two big T's, and big T, and big T. So they're all loaded. So I'm going to put in the genotypes. I'll discuss them, and you'll have to write them down from what you hear for the phenotypes. So right here I get big T, little t. Then I come along here, and I get big T, little t. And so I went big T, and big T, little t. And then when you see for the next boxes, I get the same thing. So if I'm looking at this, the only possibility is it's a 100% chance of tall, 100% chance of heterozygous. There's 0% chance of short, and that means 0% chance of homozygous recessive, and also a 0% chance of homozygous dominant. Now we do the same thing here, and this time I get big T, and I tab along there, and I get big T, little t. And I'm going to do the same thing here, and I'll use a little cut and paste action. And so big T, little t. And by convention, if we have a big and small letter, we always put the big letter first, and then I have big T, and big T, little t for that. So in this case, there's a 50% chance of homozygous dominant, which means tall, and there's a 50% chance of heterozygous, and it will be tall as well. It's 100% chance of tall, 0% chance of short. Now our last one, we go cruising along here, and we get big T, get big T, little t, we get big T, little t, and we get little t. So this is the most interesting one. So homozygous dominant, 1 in 4, 25% chance of that. 2 of the 4 boxes are heterozygous. Those would be tall as well, 50% chance of the heterozygous, and a 25% chance of homozygous recessive. So what that's saying is there's a 3/4 chance of tall, 1/4 chance of short. These 3 boxes code for tall. This box codes for short. So make sure you understand genotypes. This is showing the genotypes. We interpret them, and we can work out the phenotypes. Notice these 3 boxes are tall. This box is short, and we can say it either as a ratio out of 4, or we can say it as a percentage. So we can go 75% here, 25% there, or 3 out of 4 here, and 1 out of 4. Notice this is the genotype of the parents, where the chromosomes are segregated to make sex cells, and the same here. Inside these boxes, that's where we're looking at the possibilities for the offspring, the next generation. These are called monohybrid crosses because they involve one trait. Mono means one. Next day's lesson, we're going to look at dihybrid crosses, which are more complicated, where we have 2 traits at once. But most of the crosses you will do in this section will be variations of monohybrid crosses. So I'm hoping that makes sense. I hope you can interpret these Punnett squares. They're a useful tool.